Hello everyone. Hopefully you have seen the messages on LMS that this is a re-recording of the lectures that I did this morning for poultry reproduction and pigs, the start of pigs, because I forgot to record both lectures, a trap for people new to this system. But so what I'm doing is I'm re-recording the lectures, and hopefully then no one is going to miss out. So this is finishing off lecture 2 for poultry welfare and behaviour. So we left off last week talking about the production measures that we often use to measure welfare and production, such as mortality, hatchability, feed conversion ratio, etc. And we talked about how if we do have a sick individual, most of the time they are euthanized instead of treating that individual, although a few of you said on the farm experiences that you've been to that some farms will try and isolate and treat the bird. So I guess it depends on what the condition is and what the prognosis is and what risk they pose to the rest of the flock. So in terms of what is normal for those production measures that I mentioned before, we often go to our management guides for the different breeds. So here you can see examples for Ross, Cobb and Highline. These are available online. So if you did want any more information, then you can always go there. Now, just because I'm really paranoid, I'm just going to make sure that I'm recording. Yes. Okay. Okay, so to finish off this lecture, we're going to talk about production challenges for layers and the broiler industry. We're just going to pick out the main ones. So for layers, it's feather pecking and heavy poultry for the broilers. We'll start with feather pecking. So feather pecking is probably the most severe economic and animal welfare problem for the group housing of chickens. So it does happen in the broiler industry as well, but obviously because they are not living for as long, it hasn't become such an issue in that industry. I've given you a reference at the bottom of this slide for a very comprehensive review on this issue. It's around 16 to 20 pages, but if you do want more information, then I suggest you go look there. They summarise the latest research really well. So when we talk about feather pecking, it's been divided into 5 different categories, and Savory did this in 95 and it's still relevant today. So we have aggressive pecking, which is a normal behaviour, and that is when you have a dominant hen trying to establish her position in the hierarchy. Then we have feather pecking without the removal of feathers, and this is also a normal behaviour and is more associated with being inquisitive about other birds, and it doesn't cause any harm or stress to the recipient. And then we have feather pulling in denuded areas, tissue pecking in denuded areas, and vent pecking, which are abnormal behaviours, and they do cause stress to the recipient. So vent pecking is pecking around the cloaca, and that usually happens when birds are coming into lay. In terms of, I should say that feather pecking and cannibalism does occur in both cage and non-cage production systems, but the occurrence is more common in non-cage housing. Now that's because well, most likely because the birds are being housed in larger social groups. So that's potentially a greater stress on them. And also the feather pecker is more likely to run into a number of different birds than, say, if they were being housed in a small social group. So that's why it tends to be more common in non-cage housing. In terms of the main theories behind feather pecking, there's a few. The first is that it could be redirected foraging behaviour. So going back to the fact that they're housed in larger groups and in a higher stocking density than perhaps they would be if they were out in the wild on the jungle floor. It's possible that their environment, even if it is a barn or a free-range system, is not causing enough stimuli for them to perform that foraging behaviour. They could then become frustrated and start to try and do it on things that they shouldn't be, such as other birds. Now, this may also be associated with how they're reared. So while they're young, if they're not provided with stimuli that allows them to forage, then that could perhaps have a great impact on them later in life. Another possibility is stress. So quite a general one, but I guess stress can be the cause of a lot of problems that we see. So whether it's in the social group or in the environment, or the fact that they have to be transported and handled, certainly it's been shown that feather pecking birds are more fearful. And then once the feather pecking behaviour starts, the recipient also becomes stressed. And even if it does happen in a cage system, you will see the birds that are next door to the feather pecking event will also become stressed. So I guess it's most likely to occur when you've had a bird that perhaps hasn't had the stimuli it needs early in the rearing phase to perform the foraging behaviour, and then a certain stress occurs to it later in life in its adult phase, and then that's when we get feather pecking behaviour. Nutrition and feeding can certainly be a contributing factor, although less likely in modern production. So as I've said before, with our diets, we know exactly what amino acids, how much energy, how much fat, how much fibre, minerals, vitamins, etc. that those birds need at every stage that we really have perfected the diets. But if you do have a feather pecking event in your flock, it is a good idea to go and check diet formulations and make sure that there hasn't been an out of feed event. In terms of the control strategies for feather pecking, we've talked about beak trimming in the hatchery processing slide. So that at the moment is probably the most common way to try and control feather pecking, but obviously it's not ideal. There's a few ethical concerns with that process. Genetic selection to select for birds that have a tendency not to do that behaviour, and finding ways to encourage foraging at rearing, and also giving the birds access to perches. So the access to perches is probably more to allow an option for birds to escape a feather pecker. So now we'll move on to heavy poultry. So before I get into this slide, I just wanted to mention a couple of comments that came up in the workshop. So a few people talked about hormones being fed to broiler birds. I just wanted to clarify that yes, estrogens did used to be fed to broiler birds in the 1950s. They were also fed to cattle, and the idea behind that was to try and promote protein deposition. It kind of backfired in the poultry industry in that it promoted fat deposition. So it was stopped in the 1960s, and it's actually illegal to feed any hormones to chickens now. So when you're trying to explain to someone why broiler birds are such efficient growers and why they're so large and muscular, hopefully from the lectures you appreciate it's because of intensive selective breeding. So selecting birds that do grow fast and are very efficient at laying down protein, and also because we're able to control their environment so well. So most of the time they're housed in sheds where we can keep the temperature, the humidity, the risk of predators, etc. In perfect conditions for those birds, and also we have perfected their diets, so their nutrition. As I said before, we know what to feed the birds at every stage. So really, with those three things, we've set up an industry where the birds can. Where we've selected for large birds and also created an environment where all they have to worry about is putting their energy into growth. But saying that, early on in the industry, they did select very heavily on growth, and they forgot to look at leg strength and cardiovascular health, and they started to run into issues with brittle bones and issues with joints and sinews, etc. So since the 1960s, there's been a very large focus on a more holistic approach to select for birds that not only grow well, but also have good bone strength and cardiovascular fitness. And just to compare what a broiler bird looks like compared with a normal bird, hopefully you can appreciate the difference there. So even at day 42 of age or 43 of age, they're as big as a mature bird, even though their age responds to a small chick. So Caitlin, I think, asked a really good question in one of the lectures before in terms of, well, how long does it take to have an effect on the birds if we are going to start selecting for good bone strength and cardiovascular health? So this is some data from Canada, but remember the genetics for birds is the same for many different countries, so this in theory should apply for Australia as well. So on the left-hand side we have leg problems per 10,000 chickens slaughtered, and we're looking at a range over 20 years, and you can see that there's been a big reduction from 7 to less than 1 in 2018. And then if we look at ascites, so ascites is a build-up of fluid in the abdomen due to poor cardiovascular health. You can see that's a much more common problem at around 37, 38 birds per 10,000 chickens, but we've managed to get that down to under 5 as well. So we did talk about in our selective breeding slide how we can make changes relatively quickly in chickens because of their short reproductive cycle, but perhaps when I said quickly, that made it sound over a number of weeks or years, etc. But it's obviously does take quite a while to have an impact on what we see phenotypically, I guess. So it's taken around 20 years to see these improvements. And I guess that's because you have to wait, I guess, for a number of birds that show very good leg strength, cardiovascular health, as well as being good growers. So those things do take time. And this is just some more data. I've put the reference down the bottom. I haven't actually been able to find the original paper, but this is where the data comes from. So if we look at the graph in the top left, we have leg strength and live weight. Each line represents a population of birds. And you can see that these attributes are negatively correlated. So the line slopes downwards in that as leg strength increases, as live weight increases, leg strength decreases. But over a period of approximately 20 years, we have been able to move the data upwards. So leg strength in general has improved. And you can look at the other attributes as well. Okay, so that will finish, or that's the end of that lecture. So I'm just going to swap over now. Okay, so now we're going to be talking about poultry reproduction. I'm just checking that's coming up. We're still recording. Everything's okay. Okay, so these are our learning objectives. And we're going to start with the female reproductive tract. So female reproductive tract is made up of the ovary and the oviduct, and the oviduct is made up of the infundibulum, magnum, isthmus, the shell gland, also known as the uterus, vagina, and the cloaca. Now, in female poultry, the right-hand side regresses, so they only have the left-hand side of their reproductive tract. The way I remember that is I say lucky lefty. Lucky reminds with clucky. Hopefully that helps you as well. When we stretch out the reproductive tract, it's around 60 centimeters long. And hopefully that you can appreciate that the ovaries in poultry looks very different to mammalian ovaries, and we'll talk about that just now. So the ovary is made up of a number of follicles, and the follicles essentially contain the yolk. And within the yolk is our germinal disc, or our blastoderm, which is where fertilization will occur with the sperm. So, and the yolk is obviously there to provide nutrients to the growing embryo in the egg. So what happens is that the follicles will be different sizes, and the biggest one will rupture and ovulate, releasing the yolk into the infundibulum. So the infundibulum is which is what's happening here, is the only place where the yolk is exposed. So there's no egg white around it yet. So that is the place fertilization will occur if sperm was to be in the reproductive tract. Now this line that the follicle ruptures along is called the stigma. If you get any blood vessels crossing over that, then sometimes you can get a blood spot on the yolk. I don't know if anyone's experienced that, but that's what causes that. So this is just comparing a mammalian or a pig ovary we happen to have there versus a poultry ovary. So hopefully, well, in the pig one, you can see that we still have our follicles of all different sizes, and it's the largest one that ruptures. But in a mammalian one, they're releasing the egg. And in a poultry one, we have the yolk that gets released, and within the yolk is our germinal disc that gets fertilized. And hopefully you can see the one at the back is probably the largest, so that will get ovulated first, and then 23 to 26 hours later, the next one that is largest in size will get ovulated, and so on. So as I said, once the yolk is ovulated, It goes into the infundibulum, and that's where it'll get fertilized if sperm is present in the reproductive tract. And it will spend about 15 minutes in the infundibulum. It will then move along into the magnum, and the magnum is the area where the egg white gets secreted, so the albumin protein. It takes about 3 hours to get through the magnum. The magnum is sort of has these spiral ridges in it, which makes the egg rotate. And while the egg white protein is getting deposited, it sort of makes these pigtail-like structures, which are what are used to hold the yolk in suspension in the egg white. It then goes into the isthmus, and it spends about 1.5 hours in there, and that is where you get the shell membrane being deposited. So it's kind of like 2 thin layers of protein that get wrapped around the egg white, which is in this middle picture here. And then finally it goes into the uterus or the shell gland, and the shell is formed over 20 hours. So it gets a bit plumped up with some more albumin protein, although it's mostly water that plumps it up. And then you get the excretion of calcium carbonate, which then forms crystals, which join and form the shell. Around the shell, you also have a cuticle layer, which is like another thin layer of protein, and that is there to protect the egg from bacteria entering inside, or bacteria and other dirt and pathogens, etc. So you can hopefully appreciate in that bottom picture that the eggshell is porous. You still have gaps between the crystals. So you do get exchange of gases and potentially bacteria and other bits. But hopefully that cuticle layer just forms an extra layer of protection. Then the egg goes into the vagina. It gets flipped around in a process called oviposition, and then it gets expelled. So once the egg is pushed out, it's going from a very warm hen to usually a cooler environment. So that makes the contents of the egg inside contract, and you get this little air cell being formed, or air pocket. And that is usually the first bit of air that the baby bird will breathe when it starts pecking out of the shell. So then once an egg has been laid, it usually takes another 30 or 70 minutes before the next follicle is ovulated. So then if you go and add all that time up, that's why it takes around 23 to 26 hours to lay an egg. So moving on to the males now. So we spoke about how females only have their left side of their reproductive tract. The males have both sides. Another thing about males is that they don't have external genitalia. So in mammals, the genitalia is external because the sperm can't survive at body temperatures, but in birds it can. So that means that their testes and their vas deferens can be kept inside the body. The testes are still the site where sperm and male hormones are produced. Normally in mammals, once the sperm has been produced, it goes into the epididymis for storage. And then when ejaculation occurs, it travels down the vas deferens. In a bird, you do have an epididymis, but it's not a very large or important structure. The sperm is actually stored in the vas deferens, so another difference there. The papillae are sort of 2 little pimple-like structures that you can see in the cloaca, and that is where the vas deferens opens up into the cloaca. So if, to the trained eye, if you sort of push the vent back, you might be able to see those two little pimple-like structures. Now, the volume of sperm that a chicken produces is around 0.5 ml, so not an overly impressive amount, but it does have a very strong fertilizing power, so it's like super sperm. So it can fertilize for up to, well, it can fertilize for many days or weeks. So how does fertilization occur? So mating in birds happens via a cloacal kiss. So essentially, the male and female line up their cloaca and there's an exchange of goods. So the sperm, or in the female, they actually have a sperm gland or a little pocket where they can store sperm, because remember that sperm has fertilizing power for a number of days to weeks. So they can, the female can store the sperm and use it for a number of ovulations. So usually you only need one mating to fertilize 6 or so eggs. And remember fertilization, so the sperm still has to travel all the way up to the infundibulum where the yolk is not covered by egg white. So the sperm can remain in the oviduct for 2 to 3 weeks after mating, and it has full fertilizing capacity for about 6 days, and then after that it will decrease, but it can still fertilize. So 10th day it's got 50% ability, 19th day 15% ability. The laying cycle, we've touched on this a little bit in our previous lectures, but poultry lay eggs in clutches. So that's a group of eggs over a number of consecutive days. Obviously in production we've tried to increase the clutch length, but normally it's around 6 to 10 eggs, and it's an interval of about 23 to 26 hours between eggs. So because that it can sort of go beyond that 24 hours, you often get a bit of a lag from the day before. And when you get to a point where where the bird might be ovulating at around 3 or 4 p.m. in the afternoon, because the female's reproductive tract is light sensitive, they won't ovulate and they'll hold off until the next day. And once an egg's been laid, takes 30 to 70 minutes before the next yolk is ovulated. Does artificial insemination have a place in poultry production? Not really so much with chickens. That's usually used for specialized breeding, but it is used in turkeys. So turkeys, because we've obviously selectively bred them to be very big, muscular birds, they also have a low libido. So that means that they do need assistance with the mating side of things. So yeah, AI is normally used in turkeys, but not so much chickens. So incubation, we talked about this in when we talked about hatcheries, but if the bird's going to incubate the eggs, then that's when she becomes broody. So she stops ovulating because we get an increase in prolactin hormone, which has an effect on the gonadotrophin hormones. So you don't get any stimulation of the follicles to ovulate. That hormone is also associated with milk production in mammals, but obviously not so much, or not at all, in birds. So when the hen becomes broody, her main focus is to keep those eggs at the correct temperature. So sexing has been put into this lecture, I guess because we're talking about boys and girls, but sexing is more related to hatchery management, and we spoke about that because we obviously have surplus males for breeder farms, and certainly do have surplus males for the layer industry because we only need females. We spoke about colour sexing when we spoke about hatchery management, but there are other ways to sex chickens. Oh, sorry, in addition to colour sexing, we also talked about the technology where we're able to colour sex in ovum using a special camera. But the other ways that you can sex birds is vent or cloacal sexing. So that, once again, you have to be skilled at this, but you retract the vent back and you should be able to see the two little pimples where the vas deferens is exiting the cloaca, and that will confirm it's a male. Obviously a female won't have that. Machine sexing is another possibility. It's not used anymore because it is quite invasive. It involves inserting a blunt telescope containing a light into the cloaca of the bird to then identify either the ovary or testes. Feather sexing, once again, is where we've bred birds. So the males have slow growing feathers. So if you look at the picture down the bottom, the primary flight feathers are the feathers at the bottom of the wing, and the coverts are sort of the fluffy bits that cover the primary feathers. In males, the primary feathers are slower growing, so then they tend to be the same length as the coverts. In females, the primary feathers are faster growing, so then they tend to be longer than the covert feathers. So that's another possibility, but obviously still takes longer than looking at the colour. So colour is obviously what we discussed before, when the males tend to be yellow and the females tend to be brown. What makes a good egg? This came up a bit in the workshops last week. Size was definitely important to a lot of you when purchasing your eggs at the supermarket. So this table is just showing you the market share of the different egg weights. How do you get big eggs? It's usually related to the maturity of the bird. So a young maturing pullet that's just coming into lay will lay smaller eggs than a more mature hen who's been laying for a longer period of time. Interesting, the larger eggs tend to be paler, and that's because the same amount of pigment gets excreted into the eggshell. So obviously if it's a smaller egg, there's less surface area for that pigment to cover than if it was a larger egg. And then obviously internal quality and external quality are important factors as well. So this is the anatomy of an egg. So if we start in the center, looking at the yolk, hopefully you can appreciate there's a lighter bit and a darker bit. So the lighter bit is more heavy in protein, and that's usually laid down at night, and then the darker bit is more heavy in fat, and that's usually laid down during the day. In the middle we have the latebra, which is then connected to the nucleus of panda, and then we have the blastoderm here. So this area, the nucleus of panda, is where the embryo would grow if that egg was to be fertilized. The membrane that surrounds the yolk is called the vitelline membrane. And then we have our calciferous layer and our chalaza, which are our ponytail-like structures that suspend the yolk in the center of the egg. Then we've got our albumin, which is our egg white. So we've got an inner and an outer layer. And then we have our shell membrane, which remember is laid down by the isthmus. And then we have the shell. And then on the outside of the shell, we have the cuticle layer, which is that barrier of protection. We also have the air space as well. Now, what happens to an egg over time? So when an egg is laid, the yolk will be nice and round and firm. As the egg ages, what happens is the yolk absorbs water from the albumin, which is what's supposed to happen if we were growing an embryo. That's what would happen to support it. As that happens, the yolk increases in size because it's taking on more water, which means it stretches the vitelline membrane, and therefore your yolk becomes larger and flatter. So that vitelline membrane gets stretched, and so that's why you get the change in shape to the yolk. At the same time, because you can get exchange of gases over the shell, you get an increased loss of carbon dioxide from inside the egg, and that gets replaced with oxygen. So a loss of carbon dioxide will create a more alkaline environment or a higher pH. And that change in pH will denature the proteins in the egg white, which means that it becomes less structured and more watery. The higher alkaline environment also affects the taste of the egg white as well. So a few more factors that affect internal quality: disease. So Newcastle disease and infectious bronchitis in particular can have an effect on the quality of the eggs. Age, we just talked about. As the egg ages, the albumin gets more alkaline, which affects flavour, and it also gets more watery. The temperature, so high temperatures can decrease the quality of eggs. So eggs should be stored at 15 degrees. Saying that though, you don't really want to be changing the temperature too suddenly with eggs. So you don't want to be going from the environment that they're laid in, which could be quite warm depending on the setup, into less than 15 degrees quite suddenly, because then the contents of the egg contracts and it can draw anything that's on the outside of the shell in, which could potentially be bacteria. And then we have humidity. So a high humidity is good because it prevents weight losses from the egg, which keeps the albumin fresher for longer. Any rough handling will affect the internal quality and storage. So if you're storing it with smelly foods, then the eggs can tend to take on the smell of what they're being stored with. So then we get external quality of the eggs. So around 10% of eggs are downgraded because of shell quality issues. So I'll let you guys go through that slide at your own pace. Hopefully it's pretty straightforward, but certainly ask me if you have any queries. And then we have processing of the eggs. So the eggs are collected at least once daily. Hopefully they will be laid in nest boxes and not on the floor. If any are cracked or soiled, then they usually go to waste. Sometimes farms will pass eggs over a light so the operator can see inside the egg for any defects, and that's called candling. They're stamped with a unique code and then weighed and sorted into different sizes. Egg washing is an interesting one. So according to the Australian and New Zealand food safety codes, you don't have to wash eggs, but you can't sell a dirty egg. So the concern with eggs, I guess, is Salmonella typhimurium, which is found in the gastrointestinal tract of the bird. And then obviously, since the egg is coming out of the same spot that excrement comes out of, then there's a high chance that eggs could have Salmonella on the shell. Even if you can't see any feces on the eggshell, it doesn't mean that it's not there. And the concern, I guess, is that if you don't cook your eggs properly, if you crack them open and the contents of the egg comes into contact with the outside shell, then I guess and that can happen when you're breaking eggs, there's a risk then that you're getting transfer of bacteria. And then if you don't cook it properly, you're not going to kill that bacteria. So to try and minimize that risk, some farms will egg wash. So on small farms, it's probably more common for them to do a dry brushing. So remember, they're never cooling the eggs rapidly, and then they're usually brushing them to remove any excess dirt or feces. If there's a particularly dirty spot, then you can use a damp cloth, but you have to be very careful with the product that you use. So the water and the product, or any product you put on, has to be within the standards. Large farms will commonly wash their eggs, so usually they're washed at around the 40-degree mark. And that's to sort of minimize any risk of the egg contracting and drawing in anything from the outside in. The sanitizer you use has to have a pH greater than 10.5 and has to be approved. Obviously, if you are going to wash your eggs, you have to document every stage, and you document what you've used. You cannot let those eggs stand or soak in water. If you do, it's going to start to absorb. You're going to get transfer across the shell. And so then obviously drying the eggs is extremely important as well. Now, so hopefully you guys will know that you should be eating the egg on the right-hand side versus the left-hand side, although you obviously don't want it sitting in water for too long. And that's because of our air cell. So remember, over time, the egg is losing carbon dioxide and gaining oxygen, which means our air cell is getting bigger. So as the egg gets older, it's more likely to float. So even when you've got sort of oldish eggs, if you put them in the glass and they sink but then sort of start to sit up, then that's suggesting it's on its way. And that's all associated with the air cell depth. Now, what I would like to do is share a video with you guys that really nicely explains the formation of an egg in the reproductive tract of a female. So bear with me while I bring that up.
This presentation is...
And I'm just going to fast forward it a bit. Oh. Okay, now I'm going to share my screen.
The wall, just to the left of the spinal column. The oviduct begins at the ovary, folds back and forth upon itself, and leaves the hen's body through the vent, just below the tail. The ovary and the oviduct occupy a surprisingly small space within the body of the hen, only a few cubic inches. But when the oviduct is stretched out, it's nearly 2 feet long and has 5 distinct sections: the infundibulum, the magnum, the isthmus, the shell gland, and the vagina. When a hen is actively laying, nutrients from the food she eats are converted into the building blocks of egg yolk. These building blocks, 1/3 protein, 1/3 fat, and 1/3 water, are then carried by the bloodstream from the liver to the ovary. In the ovary, tiny tissue bags called follicles fill with yolk and grow. The largest follicle on the ovary will release the yolk of the egg the hen will lay tomorrow, while the next largest will produce the next day's yolk, and the next largest will yield the next day's yolk, and so on. In 1 to 2 weeks, a follicle grows from less than 1 millimeter in diameter to the mature size of 25 millimeters. When a yolk matures, the follicle ruptures along a line relatively free from blood vessels, the stigma, and the yolk is released. If any blood vessels cross the stigma, a drop of blood may spot the yolk as it is released from the follicle. Called the infundibulum, the funnel-shaped upper end of the oviduct envelops the ovary and catches the most mature follicle as it reaches maturation and ovulates. Then the yolk embarks on a 24-hour journey down the oviduct. When the yolk emerges from the follicle and moves into the upper part of the infundibulum, it's the only time in its progress when it is not covered by a layer of albumin. Fertilization, if it is to occur, will take place here. Some bacterial pathogens, such as Salmonella enteritidis, are able to colonize the reproductive tracts of infected hens. If these bacteria become associated with the developing egg as it passes along the tract, and before it is surrounded by a shell, they can cause disease in a human consumer of the contaminated yolk or albumin. The yolk spends about 15 minutes in the infundibulum before it passes into the magnum. In the magnum, over a period of about 3 hours, it will be covered by a dense, shock-absorbing layer of albumin, or egg white. As the albumin forms around the yolk, spiral ridges which run the length of the magnum cause the yolk to spin like a bullet in a rifle barrel. This spinning twists the protein fibers in the albumin just in front of and just behind the yolk, and makes 2 pigtail-like structures called the chalaza. The chalaza keep the yolk suspended in the center of the albumin and ultimately prevent it from moving around inside the egg. The magnum gives way to the next section of the oviduct, the isthmus. Here, the shell membranes are deposited. These thin layers of protein wrap loosely around the albumin covering the yolk. It is as though the yolk and its layer of albumin are a blob of jello wrapped with 2 sheets of cellophane. The process does not result in a smooth egg-shaped structure. In fact, an egg leaving the isthmus probably looks more like a prune than a plum. The partially formed egg then enters the shell gland. Here, over the next 20 hours, the shell will form. First, a thin albumin is secreted. This thin albumin is mostly water, and it moves by osmosis through the two shell membranes into the highly concentrated thick albumin surrounding the yolk. This plumps the egg into a normal shape and stretches the shell membranes tight around it. Next, a highly concentrated solution of calcium carbonate is secreted by the shell gland, and crystals of calcite form and grow on the outer shell membrane. As the crystals expand, they grow into one another to form a solid shell. Very tiny spaces left in between the crystals leave pores in the shell. Lastly, a special protein solution, called the cuticle, is deposited onto the eggshell. Gas can pass through the proteinaceous cuticle and through the pores in the shell, but the two layers protect the egg from harmful bacteria. Finally, in a process called oviposition, the egg flips end over end. This occurs through contractions of the uterus, synchronized with relaxation of the muscular vagina, and pushes the egg out of the hen's body. An important part of the egg does not form until after it is laid. When an egg is laid, it fills the shell. However, a hen's body temperature is 106 degrees Fahrenheit, and eggs are generally laid into environments that are 20 to 40 degrees cooler. As the egg cools, the inner portion contracts and forms an air cell between the two shell membranes. A chick would puncture and breathe through the air in this cell before hatching. The fully formed egg now begins another journey. If it has been fertilized, it will go to a hatchery. If not, it will go to one of two places: an egg processing and packaging plant for distribution to grocery stores, or to an egg breaking plant where, once processed, the egg will be included in one of countless food products, such as mayonnaise, cake mixes, or salad dressing.
Okay, so hopefully that helps consolidate things around the female reproductive tract. I just want to make one point. The salmonella they mentioned in that video was Salmonella enteritidis. And that, luckily enough, is not present in Western Australia, but they did have an outbreak of it on the East Coast. So that is different to Salmonella typhimurium. So Salmonella enteritidis obviously lives in the reproductive tract of the bird and can infect the internal yolk and white. Salmonella typhimurium is usually lives in the gastrointestinal tract of the bird, and that's what you get on the outside of eggshells. So something to just clarify there. Okay, so I apologise once again that I didn't record the initial lecture, but hopefully that was a more polished version anyway. And I will go ahead and record the pig lecture now as well.